So welcome back, and we'll pick up our discussion with autoimmune disease affecting the joints. So we'll break it down into these different sections. The intro to autoimmune disease, review again of eicosanoids and inflammation, cytokines and inflammation, and then we'll take a look at these autoantibodies and anti-nuclear antibodies and other blood markers that are found in autoimmune diseases, just so we are clear on what kind of lab work you might see with these patients. And then we'll look at some common drugs that are used to manage autoimmune disease, and then we'll move into the nutrition and natural medicine management of autoimmune disease in terms of the anti-inflammatory diet and supplements, things that can help to tame nuclear factor kappa beta and tumor necrosis factor alpha, things that might modulate the immune system in a positive way, how you can help prevent joint cartilage damage, and other lifestyle considerations that would be of importance. So let's move to the discussion. So the intro to autoimmune disease. Of course, autoimmune diseases involve an abnormal immune response to substances and tissues that are normally present in the body. So the immune system is now attacking the body's normal tissues and substances. There's usually the presence of autoantibodies and or self-reactive T cells with or without damage or pathology resulting from it. So, very often you have antibodies that are made that are attacking the tissues, a specific tissue, because it finds an antigen that it wants to attack that's actually part of the body's normal structure. And often you get these self-reactive T cells where the CD4 cell morphs into a CD17 cell, secreting interleukin 17, which produces a great deal of inflammation and upregulation of tumor necrosis factor alpha that perpetuates autoimmune disease. You're going to see, as we talk about different autoimmune diseases, and also when we talk about Crohn's disease in the gastrointestinal course, and we saw it in multiple sclerosis in the neurology section, that the CD17 cell secreting that high interleukin 17, if you can tame that, then it helps to calm down the autoimmune disease in a significant way. And there seems to be some natural things that can help to do that. So in an autoimmune disease, the disease may be restricted to a certain organ, so may just be the antibodies might just be attacking the thyroid gland, let's say, or maybe a particular type of tissue like the alpha-3 subunit type 4 collagen, which is widespread, that you would see in Goodpasture's disease, where the antibodies are attacking the basement membrane, wherever that collagen is in the lungs and in the kidneys and so on, leading to bleeding from the lungs and resulting in kidney failure as well. So, it has a lot of different faces when you're talking about autoimmune diseases. And so here's a list of the many of them. And on this slide, it actually shows you what the target is. The antibodies is attacking, in myasthenia gravis, the acetylcholine receptor. And in Graves' disease, the thyroid-stimulating hormone receptor. In asthma, the beta-2 adrenergic receptors. And in Addison's disease, adrenal cells are being attacked. And in vitiligo, the melanocytes are being attacked. And then you see many autoimmune diseases, you see attacking of certain aspects of the DNA or the RNA. So there are these anti-nuclear antibodies sometimes that they find that are sort of pathognomonic of certain types of autoimmune diseases when they're looking at blood tests. So we'll come back to some of that. But just once again, in autoimmune disease, you're getting an upregulation of the pro-inflammatory eicosanoids and prostaglandins, as phospholipase A2 cleaves off arachidonic acid from the phospholipids and increases its conversion to these inflammatory prostaglandins via cyclooxygenase and lipoxygenase. And again, we understand some diet and lifestyle things can be helpful, as well as non-steroidal anti-inflammatory drugs and corticosteroid drugs. So once again, you would see that arachidonic acid would be released from the phospholipid structure via cyclooxygenase. It forms many prostaglandin series 2 agents that produce inflammation, and in white blood cells, arachidonic acid via lipoxygenase becomes very inflammatory leukotriene B4. So in rheumatoid arthritis, as an example, in the synovium of the rheumatoid arthritic patient, the white blood cell count is extremely high, and those monocytes morphing into macrophages are producing copious amounts of leukotriene B4 and promoting a lot of that inflammation. It's where a lot of it's coming from. So being able to block lipoxygenase is important. So this is why I think that things like boswellia and ginger and curcumin are so important in those conditions as part of the long-term management to help decrease the patient's reliance on prednisone and other agents and biologics as well, like Humira and Remicade and these types of things. But also you want to try to minimize how much arachidonic acid there is in the cell membrane in the first place, and that's where dietary factors come in and supplementation with essential fatty acids. Remember that icosapentaenoic acid from fish oil can help to also block the release of arachidonic acid from the structure by inhibiting phospholipase A2. And then again, tumor necrosis factor alpha and the related cytokines play a huge role in autoimmune disease. So that, again, the TNF being secreted by different immune cells, macrophages, the CD17 cell, stimulating the tumor necrosis factor alpha receptor and upregulating through nuclear factor kappa beta the genes to give rise to these inflammatory interleukins and cytokines that really perpetuate the major inflammatory disease and upregulating the metallomatrix digesting enzymes that destroy joint cartilage and connective tissue. So once again, it's that nuclear factor kappa beta that sits there in the cytosol, usually minding its own business, but upon stimulation via receptor tyrosine kinase and phosphoinositol 3 kinase, stimulating it as different receptors get affected by the immune cells affecting the receptor from the outside in. And then, as we saw earlier, the macrophages then, in this inflammatory response are secreting tumor necrosis factor alpha and upregulating inflammation. And the synoviocytes are also secreting metalloproteinases that destroy the joint cartilage, all under the influence of tumor necrosis factor alpha, being secreted by various immune cells, including macrophages and the T lymphocytes, known as the CD4 cell, which in the autoimmune diseases morphs into often a CD17 cell, as we're going to see. And so they have created drugs that are called monoclonal antibodies. And of course, these drugs are really what they did originally. I think this is how they still make these drugs. Is they take the tumor necrosis factor alpha receptor, which is actually just a protein on the surface of the cell, and all proteins are made out of amino acids. So through reverse engineering, they can really figure out what the amino acid sequence is. There's different ways to determine that. I think electrophoresis possibly. And then they can build that TNF receptor, and then they can expose it to immunoglobulins originally derived from multiple myeloma patients, I believe. And in animal models, they can get that immunoglobulin to produce antibodies against the TNF receptor to destroy it. And that becomes the drug. The drug is actually an antibody that destroys the receptor. So if you destroy the receptor, it can't respond to the tumor necrosis factor alpha that's being secreted. And so they've made different monoclonal antibodies to attack different types of targets that are part of the inflammatory cascade in autoimmune diseases. So you see them here. Humira, Remicade, these are popular. Cimzia, these are all really popular and common ones. And that's basically how they're working. But remember that as you destroy these receptors, and sometimes they can't respond in a positive way, if a cancer cell is emerging or if the person's getting an infection, it's hard to recruit an attack. So that's sort of the downside of it. And also remember that the monoclonal antibody is a foreign substance to the body. So sometimes the body forms antibodies against the antibody, and the person feels very sick and unwell when the person's given a drug like that. They also use a monoclonal antibody in a type of breast cancer called HER2 positive, where the person's making overexpressing the epidermal growth factor receptor type 2 on their cell surface, so the cell is dividing at a very fast rate, and then it can become a cancer cell quite easily as genetic mistakes accumulate. So if they see the HER2 receptor is there, you can destroy the HER2 receptor with a monoclonal antibody called Herceptin, and it helps to prevent the recurrence of that type of breast cancer, but and to help contain the existing cancer. But it also produces a lot of adverse side effects. So you can't just put that in the drinking water as a widespread measure to decrease breast cancer risk across the population, because it doesn't make the person feel sick. So here you see what happens. This is the naive T cell. This would be a CD4 cell that hasn't been prompted. It's just sitting there waiting for stimulation to occur. And then the antigen presenting cells come along. The macrophages, the dendritic cells, the B lymphocytes, these are all antigen presenting cells, and they'll present an antigen. And sometimes the CD4 cell then morphs into a Th17 cell and starts to produce an autoimmune inflammatory response. And so you see that in MS, you see that in Crohn's disease, you see that in a lot of different autoimmune diseases, the prevalence of the CD17 cell that was originally just a CD4 cell minding its own business, and the person's going along and their health seems to be fine, and all of a sudden you've got this immune system attack that's going on. So it can be prompted by a virus, a bacteria, environmental toxins, pesticides, herbicides. We don't even know all the different factors that could actually prompt that cell to start to transform into this CD17 cell. And often it also, with the autoimmune disease, the CD4 cell often morphs into the Th1 version of the CD4, which upregulates a lot of inflammatory mediators that you also see in autoimmune disease as well. Whereas in allergy, you see more of the Th2 phenotype. We'll keep going. So here you see that, again, this would be the CD4 cell. Upon exposure to the antigen, usually increasing the amount of Th1 phenotype that it starts to express, where it's secreting a lot of interferon and interleukin-2 by these immune cells, promoting a lot of inflammation. Whereas if it's prompted, say, in an allergic reaction to produce more Th2, you see more of the mast cells releasing histamine, and the eosinophils are more reactive. These are things you normally see in an allergic type of response. So the CD4 cell is sort of the quarterback of all of this. And so in general, you have these antigen-presenting cells. These would be the macrophages and the dendritic cells and the B lymphocytes that are finding things that don't seem to belong in the body, and they ingest them, and then they sort of express the unusual receptors from those things they're ingesting on their cell surface, and they bring it back into the lymph tissue, and they find the CD4 cell, and the CD4 cell then responds to what it's seeing. So normally in a viral or bacterial infection, it's going to recruit the immune cells to go out and attack that particular pathogen and kill it. And then once the killing is over and everything's calming down, the CD8 cell, also known as a T regulatory cell, usually then says, okay, we have time to suppress this inflammatory response and suppress the immune system response. The fight is over. We don't need to be upregulated forever. But what happens in the autoimmune disease is that the antigen presenting cell stimulates the CD4 cell in such a way that it morphs into a CD17 cell and secretes Th17 interleukin, which then upregulates the secretion of all kinds of inflammatory interleukins and also tumor necrosis factor alpha. And that really kicks things into gear. And also, the CD4 cell tends to secrete more of the Th1 type of cytokines that also upregulate more inflammatory cytokine response. And so you get this huge pro-inflammatory response, and the T regulatory cells can't really balance it out. And so you're getting this perpetuation. So controlling what that CD4 cell does is at the heart of a lot of these autoimmune responses. And then we have this, which I had alluded to earlier, where you get viruses or bacteria sort of inducing rheumatoid arthritis by injecting their DNA or RNA into the monocyte progenitor cell. So the monocyte lineage normally will produce macrophages, as we know, but also osteoclasts and dendritic cells. So the monocyte gives rise to macrophages, dendritic cells, and to osteoclasts. And dendritic cells, remember, they're antigen-presenting cells along with the macrophages and the B lymphocytes. And the monocytes, once they come out of the bone marrow, they take up residence all over the body, in the lungs, in the kidneys. They line the synovial tissue. So when they morph and they go into bone and become osteoclasts, they go into the circulation as dendritic cells, part of the innate immune system. And when they morph, if they become infected or their DNA gets hijacked, and they now become pre-shaped, as we see here in the second line, the pre-shaping of these, the progenitor cells, makes these cells, the entire lineage, start to behave in a very unusual way. So the macrophages are upregulated, the dendritic cells are upregulated, the osteoclasts are upregulated. So you're getting inflammation and destruction of bone. You're getting abnormal antigen-presenting cells telling the immune system to maintain the inflammatory response. So these pre-shaped monocyte macrophages, osteoclasts, account for respiratory involvement and bone demineralization associated with rheumatoid arthritis and other connective tissues. So it's not just the joints are being affected. Wherever the monocytes go, all of those tissues are going to be affected. So when you're studying rheumatoid arthritis, in the beginning it doesn't make sense. Why are the joints being affected? Why are the kidneys being affected? Why are the lungs being affected? Why is there subchondral bone loss? It's because wherever the monocytes go, if they've been pre-shaped and their DNA has been hijacked in the bone marrow itself, then all of the daughter cells from that monocyte progenitor cell will all behave in an autoimmune type of abnormal way, and that makes it very difficult to treat. So in each instance, tumor necrosis factor alpha is the principal driving mechanism to upregulate the release of inflammatory hyperproliferative extracellular degrading matrix metalloproteinases, which destroy joint cartilage, and angiogenic cytokines. So blocking TNF release has been shown to suppress the inflammatory response, but it can have serious effects. So that's where those biologic treatments come in with Remicade and Humira. If you can destroy the tumor necrosis factor alpha receptors, then those cells are not responding to what those monocyte daughter cells are trying to do. So you can arrest the reaction, if you will. And also upregulating the increase of interleukin-4 has an anti-inflammatory effect. So that's nice to get more interleukin-4. And vitamin D supplementation has been shown to increase interleukin-4. It's one of the ways that vitamin D we think is helpful in MS and other autoimmune diseases, including some inflammatory bowel diseases. So in rheumatoid arthritis, TNF is over-secreted by overzealous macrophages with resulting inflammation, fibrosis of the joints and internal organs, degradation of extracellular matrix, and increased vascularization. So TNF alpha is induced by the interleukins, the CD4 cell that becomes the interleukin 17. It's being induced by that crazy CD4 morphing into the CD17 cell, which is found at high concentrations in rheumatoid arthritic patients. And inflammatory cytokines activate macrophages to trigger production of inflammatory prostaglandin 2. So it upregulates the release of arachidonic from the phospholipids to produce more type 2 prostaglandin inflammatory eicosanoids. And that's how all these things sort of tie together. This was a great review of this entire model found in the journal Arthritis Research in the year 2000. It's a fantastic review of this. So then we have anti-nuclear antibodies and other inflammatory markers. So in autoimmune disease, there's often the presence of some combination of autoantibodies, so antibodies attacking the body's tissues that can be found in the blood, and or anti-nuclear antibodies that are actually attacking a part of the DNA within the cell, and other inflammatory markers like rheumatoid factor and a high SED rate and C-reactive protein. So their presence aids in the diagnosis of the specific disorder. Sort of what that whole constellation of blood tests tells you is how the diagnosis is generally made. So with respect to antibodies and other blood markers of autoimmune disease, antibodies are proteins produced by lymphocytes, the B lymphocytes, and plasma cells. Remember, they come from the same lineage, which normally circulate in the blood to defend against foreign invaders such as bacteria, viruses, yeast, and toxins. And then you have autoantibodies. Instead of acting against a foreign invader, as normal antibodies do, these autoantibodies attack the body's own cells or a certain type of tissue. Then you have anti-nuclear antibodies. They're a unique group of autoantibodies that have the ability to attack structures in the nucleus of the cell itself. And then these anti-nuclear antibodies, you would do this test on the patient's blood sample as part of the diagnosis process to detect certain autoimmune diseases. So you have a screening test for these different autoantibodies and anti-nuclear antibodies to try to arrive at a diagnosis. So the incidence of anti-nuclear antibodies in various diseases or conditions. So you see the positive in over 95% of lupus cases, you find positive anti-nuclear antibodies. In progressive systemic sclerosis, 60 to 90% of the time. In rheumatoid arthritis, up to 30%, Sjogren's syndrome, and you see all the way down to juvenile arthritis. And there are subsets of these anti-nuclear antibody tests that are sometimes used to determine a specific autoimmune disease. So for rheumatologists may order an anti-dsDNA or an anti-Sjogren's syndrome antigens. There's different types of these autoantibodies that are found. So for instance, there's the ANCA antibodies that are found in ANCA vasculitis. So this is a type of immune response where the antibodies are attacking the neutrophils, and then so you get neutropenia, and then the neutrophils in turn start attacking blood vessels somewhere in the body, sometimes in the skin, sometimes in the kidney. You can get this ANCA vasculitis that actually causes kidney disease. And also there's the HLA-B27 antigen that's often assessed as well. So when we come back, we'll pick up the story, I think, with the HLA-B27 antigen. Let's take a break and we'll come right back.